Hello, good day, everyone. Um, Sorry, today in the morning I was unable to record the session we had. Unfortunately, I actually don't know what happened until after we finished the class. I realized that I have not been recording the session. So I decided to create the time I record the class again, and I will also record the one I promised to record for the rest part of the class. Okay, now we discuss on module 3 and still entomology, study session 1, chemistry of amino acids and protein 2. Remember in the last previous class, we talked about the chemistry of amino acid and protein 1. So under these two, we have classification of lipid section and subsection headings. So here we will be discussing basically on lipids, the classification of lipids. We'll talk about the fatty acid. We talked about eicosanoids. We talked about triacylglycerols, their compositions, classification. We talked about steroids. We talked about lipoproteins. So discussing about lipid. We define lipid as substances that are generally heterogeneous in nature. First of all, we said the word lipid was from the Greek word lipos, which means fat. And we say these molecules are heterogeneous in chemical nature and as well as their function. So by definition, we can say they are generally an organic molecules, or organic macromolecules that are relatively insoluble in water and soluble in organic solvent. Unlike the other micromolecules we talked about. We talked about carbohydrates, nucleic acid, and proteins, where we said these are macromolecules and they are a polymer of some monomeric unit. In the case of protein, we said is a polymer of amino acids. In the case of nucleic acid, we said they are polymer of nucleotide, while carbohydrates, they are polymer of monosaccharides. But lipids are not a polymer. They are not polymer. We'll see what are the compositions, what are the substances that comes together to give lipid. But basically, we can say a lipid contains a fatty acid. Either it's a fatty acid being converted to another substance, or the fatty acid form an ester with another substance to give a different classification of lipids, which we are going to shortly discuss. Now, classification of lipids. Lipids were classified into three. We have a simple lipid, we have a complex lipid, and we have a dry lipid. Now we'll see why we classify them three. Why simple differ from complex and as well differ from the dry lipid. Now, in simple lipids are esters. We explain what are esters. So they are esters of fatty acid and various alcohols. They are classified into two. We have fat and oil and waxes. Fat and oil is an ester of fatty acid and a glycerol. Usually, triacylglycerol is the major component of this fat and oil. While in waxes, we have a fatty acid ester with an alcohol rather than a glycerol. The alcohol rather than a glycerol usually used to be a very long chain. So also the fatty acids. So the basic difference between fat or oil and waxes is number one, the fatty acid in waxes are usually a very long chain fatty acids, and the alcohol is not a glycerol in waxes, while in fat and oil is glycerol. So in waxes can be something else but usually it used to be a very long chain alcohol. So you have a different alcohol found in waxes. Now, the fat and oil. Fat and oil is a typical example of a simple lipid. In this class, as we said, we have a typical of the most common, which is triacylglycerol. And the triacylglycerol can exist either in solid or in a liquid form at room temperature, depending on some certain condition, which we will talk about while talking about triacylglycerols. But basically, what are the components of this triacylglycerol? Triacylglycerol is made up of a fatty acid and a glycerol. We can see the structure of fatty acid by our left. This is a typical structure of a fatty acid where we have a hydrocarbon and a carboxyl group. And next to it, we have a structure of a glycerol. We say it is an alcohol. It has a 3-OH group and a 3-carbon molecule having a 3-OH group. Each OH group at 1 carbon atom, a particular carbon atom at carbon 1, carbon 2, and carbon 3. How the esters are formed is we said is the reaction between this hydroxyl group and the carboxyl carbon of the fatty acid, leading to formation of water. So water is released and lead to formation of this bond called an ester bond. So on carbon 1, so also when it happened on carbon 1, we call it monoacylglycerol. When this esterification occurs on carbon 2 and carbon 1, it's regarded as diacylglycerol. But when this esterification occurs in all the carbon, the 3 carbons, it is called triacylglycerol. Triacylglycerol is referred as the neutral lipids because it does not have any charge. And of course, it is the one of the core lipids that are highly hydrophobic in nature. They do not interact with water. There is no any group that is hydrophilic that might make the molecule to be a bit soluble. So while in the case of waxes, I've already explained, we said we have a very long chain fatty acids, and we have a very long chain alcohol. So the esters between this long chain fatty acid and a long chain alcohol rather than glycerol is what we call waxes. Of course, in the case of triacylglycerol, I've forgotten to mention one of the main functions of triacylglycerol is used as an energy source in animals. So it is stored in the body for energy source. So also waxes. Waxes can be stored by plants and some insects that also serve as energy source as well as a protective cover. We can see that example of waxes include lanolin, beeswax, whale's palm oil. I've given the picture of beeswax for those of you that were not familiar with beeswax, and we are very much familiar with this thing and this net, and this is basically made of wax. Now the next class of lipid is complex lipid. The complex lipid are also esters of fatty acids with alcohol, but in this case they have an additional group called a prosthetic group. This prosthetic group can be a phosphate group, it can be a nitrogenous base, it can be a carbohydrate, it can be a protein. If it is a phosphate group, the lipids are called phospholipids. If it is a carbohydrate, they are called glycolipids. If it is a protein, they are called lipoproteins. So shortly, we are going to see some of these. What are those things? And in addition, we have another discussion in 102 slide where we'll talk about lipoproteins. So in phospholipid, what happened in phospholipid is, just remember the explanation we gave with regard to triacylglycerols. In triacylglycerols, we say we have 3 fatty acids, and these fatty acids form an ester with the the hydroxyl carbon, the 3-hydroxyl carbon of triacylglycerol. In the case of phospholipid, we have the fatty acid and the glycerol. So the two fatty acids were attached to carbon 1 or esterified to carbon 1 and carbon 2. While at carbon 3, we don't have a fatty acid. Rather, we have a phosphate group. So when you have a fatty acid, two fatty acid, and one phosphate at carbon 3, we call it phosphatidic acid. We call it phosphatidic acid. So before that explanation, phospholipids are classified into two. We have glycerophospholipid and sphingophospholipid. In glycerophospholipids is the phospholipid that contain the fatty acid and the glycerol, or the fatty acid were esterified or attached to an alcohol, which is glycerol. So we have here, we can see the fatty acid. The fatty acid esterified to glycerol. And at carbon 3, we said instead of fatty acid, a phosphate group is attached to carbon 3. So we can see the phosphate group that was attached here. So another group can be attached. We talked about that. But before the other group is attached, let's just assume that a phosphate group, and we said it is called phosphatidic acid. And phosphatidic acid is a precursor for the synthesis of any phospholipid. I explain some of those groups that are usually attached to this group while discussing about the membrane lipids. An example of phospholipid as one of the membrane lipid, where we made a mention of different groups, and I think I also put their own structures like ethanolamine, choline, serine, inositol, etc. So the other classification we call sphingolipid. Sphingolipid in this case is a fatty acid ester with an alcohol, but in this case is a sphingosine, not a glycerol. Now we can see how the sphingosine look like. In the next slide, we'll see a structure of a sphingosine. And then on the last carbon, we have the phosphate group. And then any other thing can be added, which can be choline, serine, ethanolamine, as in the case of glycerophospholipids. So, like I said, the glycerophospholipid, we have example of phosphatidyl choline. When choline is attached to the phosphate group, we have phosphatidyl ethanolamine. When ethanolamine is attached to the phosphate group of the phosphatidic acid, we have phosphatidyl serine. We have phosphatidyl inositol. We have plasmalogens and cardiolipins. While in the case of sphingosine or sphingolipids or sphingophospholipid, we have sphingomyelin, which we also discussed while talking about the membrane lipids. So basically, earlier in the morning, I also explained the structure, and this is the phosphatidic acid I was talking about, where at carbon 1, we have a fatty acid esterified. At carbon 2, we have another fatty acid, while at carbon 3, we have a phosphate group, and we call this a phosphatidic acid. Now, to this phosphate group, many groups can be added, which I said could be choline, ethanolamine. When choline is added, it is called phosphatidyl choline, as in this case. Phosphatidyl choline is also called lecithin. Here we have phosphatidyl ethanolamine, where ethanolamine is added. You can see the added group in red. So phosphatidyl ethanolamine, it is called also cephalin. And here we have phosphatidyl inositol. This is inositol. And then we have phosphatidyl serine. This is the serine that was added in the listen. But in the case of sphingophospholipid, like I said, sphingophospholipid does not contain glycerol. Rather, it contains what? It contains a sphingosine backbone. So we have the sphingosine backbone here. When you look properly, it's not a glycerol. So from here out to here, we call it the sphingosine, and we can see this carbon, which is supposed to be the carbon 2 as when compared to glycerol, we have a fatty acid attached to. While the last carbon, which when compared to glycerol, is supposed to be carbon 3, now we have the phosphate group attached. So on the phosphate group, we can have any of this group added. Here we have a plasmalogen where where ethanolamine is added to form phosphatidyl ethanolamine. So the next class of lipids are called glycolipids. The glycolipids, they are just like the sphingophospholipid. They do not also contain glycerol. Rather, they have a sphingosine backbone. And we have the fatty acid attached. And then the next, at the last carbon, where we have the phosphate group, as in the case of phospholipid. Here we have a sugar, a carbohydrate attached to it. When just one sugar like galactose is attached to that carbon, it is called cerebroside. But when we have an oligosaccharide, more than one carbohydrate or more than one monosaccharide attached by a glycosidic link is attached to that carbon, where the phosphate group is supposed to be, as in the case of phospholipid, we call it ganglioside. Now, the next class of complex lipids are lipoproteins. In the case of lipoproteins, we have a lipid attached to a protein. So the protein now serves as a prosthetic group, and they are classified into 4. We have chylomicrons, very low density lipoproteins, and high density lipoproteins. These classifications basically based on the density of the complex that is formed. We'll talk about lipoproteins shortly. I will have a short video also where I will talk about lipoproteins. Drive lipids. Now, what are drive lipids? Drive lipids are lipids that are obtained from hydrolysis of the simple lipid and the complex lipid. Remember, we said the simple lipid is made of an ester of fatty acid and a glycerol. So when we have a hydrolysis, we will have the individual component which forms the simple lipid. And what are these individual components? Are the fatty acid and the glycerol. But remember, we talked about the formation of triacylglycerol. When we have the first fatty acid attached to glycerol, we are going to have monoacylglycerol. So hydrolysis can also lead to production of, as one of the product, monoacylglycerol. It can also form a diacylglycerol. So these are some of the hydrolytic products of hydrolysis of. And in addition to that also, you can have a fatty acid being used also by this, most especially these polyunsaturated fatty acids that are being used to produce quite a number of structures that have some bioactive properties. Some of them are steroids, some of them are cholesterols, some of them form vitamin A, vitamin D, all these lipid-soluble vitamins, eicosanoids. So basically, they are formed from the fatty acids. Usually polyunsaturated fatty acids tend to become the precursors to which these complex structures are formed. And these complex structures have various biological functions. So they are basically classified under dry lipids. So we talked about the functions of lipids. Basically, we said it's formed a major component or one of the component of dietary proteins. And when we talked about that, we say, why do we have them as part of the food? We said it improves taste and it improves or it gives palatability, making you to have the appetite to eat the food, make the food flow to eat the food and enjoy it. And we said some of these lipids serve as an energy storage. We'll talk about triacylglycerol. We'll talk about it, where the triacylglycerol serve as one of the major source of energy storage in the body. Lipids also serve as a component of cell membrane. We talked about phospholipid and how phospholipid form the major component even in the formation of phospholipid. So I don't think you need much explanation on that. And we talked about transportation of substance in and out of the cell, how this phospholipid or how this membrane lipid facilitate the movement of most especially the water-insoluble substances in and out of the cell. I just talked about some of those lipids or polyunsaturated fatty acids serving as the precursors in the synthesis of fat-soluble vitamins like vitamin A, D, E, and K. We also talked about some of them serving as the precursors of some metabolic regulators like both endocrine and exocrine hormones like testosterone, androgen, progesterone, etc. And we said that they can also form a kind of protections to organs. You can see most of the organs in the body, kidney, liver, and so many of them have some parts that are deposited that cover these organs. So for those of you that have ever slaughtered an animal, they will discover some of those parts that was surrounded some of these organs. They are basically giving them protection. And I talked about also maintaining the shape and the smoothness of the body appearance. So this is basically about lipids. I want to create a different video for the next. So please, you have to watch this class. If you have any question, you raise the question in the platform or under the video where I uploaded it. Please try and ask your question there. Just drop your question there. I'll respond to it. When you are very satisfied with what is here, then please you should like the video so that let me know the response, whether you guys are really getting the message. Thank you very much.